All right, so let's dive into chapter 9 on energy and metabolism. All right, yeah, I know. I remember doing energy metabolism. It was not my favorite, for sure. All right, so we're going to talk about metabolic reactions. And so we have these metabolic reactions with the biochemical reactions that go from the beginning to the end. And then we have stuff that helps these reactions to occur. And these substances that help reactions to occur are called enzymes. Okay? What did I tell you about enzymes? Enzymes are mostly made up of what? Proteins. Yeah, always remember enzymes are proteins. And they have these cool three-dimensional shapes that help to help them so that they can then bind to the substrate for these enzymatic reactions to occur. Okay? So we have some intermediates, the little compounds that are formed along the pathway, and then we have two terms, anabolic and catabolic. Let me give you a hint on how I could always remember the difference between the two. Anabolic, to build up, catabolic, break down. Okay? So we're constantly building up, breaking down, building up, breaking down. The way I could always remember the difference between the two is when I saw catabolic, I always thought of cannibalic, like cannibalism. Y'all know what cannibals are? They break things apart and eat, cannibals eat people, right? For whatever reason, I think cannibal, catabolic. I think of them as the same thing, breaking it down. I know that sounds kind of morbid, but the words almost look the same to me. All right, so let's use some examples. All right, so we've talked about this before. Proteins are broken apart into what? Proteins are made up of many amino acids linked together. So in a catabolic reaction, you're breaking apart proteins into amino acids. Carbohydrates are made of many what? Glucose molecules linked, like starch, many glucose molecules linked together. So a catabolic reaction that breaks down starch will break apart starch into glucose. Fats are broken apart into what? Fats are made up of, a triglyceride is made up of glycerol and 3 fatty acids. So catabolism, you're breaking apart those bonds and you're breaking apart fatty acids and glycerol. Well, then our cells even go further, right, in these reactions. So they'll take things like glucose, fatty acids, and break them apart and use the energy in breaking the bonds to make ATP. Right? So in that process, CO2 is released. Also, we'll see water, ammonia. So all these are components of breaking down of catabolic reactions. Versus the opposite then of catabolism is what? Anabolism, building up. So amino acids are linked together with peptide bonds to form proteins. Glycerol, 3 fatty acids are linked together to make triglycerides, right? And then many glucose molecules are linked together to make glycogen. Yeah. So it's kind of cool how that works. Okay. So these are just showing you the difference between catabolic reactions and anabolic reactions. And remember, all these substrates and the catabolic reactions, the simplest things, glucose, the fatty acids, the glycerol, the amino acids can all be used to make ATP in catabolic reaction.
What's ATP? What is ATP?
I'm so glad you asked what ATP is. We see that little abbreviation ATP, adenosine triphosphate. All right, so what does that mean? Triphosphate. 3 phosphates linked together, yeah, with a sugar. So we're using the energy from these energy substrates to phosphorylate, to add the phosphate groups to ADP. What is ADP? Adenosine diphosphate, which would be what? 2-phosphate. Or we could even do AMP. What is AMP? Adenosine monophosphate, which would have 1 phosphate group, right? So in this process, we're taking the energy from these energy-yielding substrates, breaking the bonds, and using the energy with electrons to add the phosphate groups from ADP to make ATP, putting them back together. So we're breaking it, building it, breaking it, and building it. So what do we even need ATP for? Lots of stuff. What kind of stuff do we need ATP for in the cell? We know it's energy, right? The energy the cell uses to do stuff, right? Will muscles contract without ATP? No. So we have to have ATP for muscle contractions. What about nerve impulses? Do they need ATP in order to work? For neurons? Yeah. What about pumping ions? You think that's going to take energy and ATP to pump ions outside the cell? Yeah, of course. So, absolutely. So, pumping ions, muscle contractions, nerve impulses, they all need ATP. All right. So you can see those catabolic reactions: proteins broken into amino acids, carbohydrates into monosaccharides, triglycerides broken down to through fatty acids, glycerol, and all of these things then will be used. And you can see acetyl-CoA. We'll talk a little bit more about acetyl-CoA in a little bit. And all this process of making ATP. All right. So adenosine triphosphate. All right. So you ready? So list the catabolic pathways, list the anabolic pathways, and what is the energy currency the cell uses to do stuff like muscle contractions, nerve impulses, and pumping ions. What does a cell need to do that stuff? All right, so on page 286, catabolic reactions, anabolic reactions. You guys get to fizz, you'll be an old pro with this, right? You have to list this stuff out pretty quickly in that class. So list the catabolic pathways, list the anabolic pathways, and what is the energy currency of the cell? It's on page 286, 287. So list the catabolic reactions, list the anabolic reactions or pathways, and what is the energy currency of the cell? What does the cell need to pump ions, muscle contractions, nerve impulses? Yeah. What do we call the energy currency of a cell? All right, y'all ready to go to the next thing? Y'all ready? Okay, here we go. And then we see the terms oxidation reduction. And sometimes people get a little bit confused on this one because what we're doing in these reactions is we're shuttling the energy carried in these electrons, remember, to add the phosphate groups back together. Does that make sense? So we're gathering the energy and we break these substrates to carry that energy in electrons so that we can add put the phosphate groups, link the phosphate groups back together. Okay, so we use reactions called oxidation reactions. So if it oxidizes, it means it gains the oxygen, loses a hydrogen, and loses an electron. Versus reduction is it gains the electron loses oxygen and gains hydrogen. So we have these coenzymes, which are energy shuttles, right? And these coenzymes are called NAD plus and FAD plus. And an easy way to remember where they came from is NAD is a B vitamin called niacin. You see N for niacin. So NAD is the B vitamin niacin. And FAD plus is the B vitamin riboflavin. Okay? So you see F for riboflavin. Always think of flavin in the middle, right? Riboflavin. So NAD is from what? Niacin. FAD is for riboflavin. All right, so what if we don't get these B vitamins in our diet? You always hear people talk about that a lot. If I don't get enough B vitamins, I feel very tired. Are they giving you any kilocalories? There's no kilocalories in vitamins, but they're shuttling electrons for these energy reactions. So are these energy reactions going to occur if you don't have enough niacin or riboflavin? No. So we have to have these B vitamins because they act as coenzyme in energy reactions. Okay. So all the B vitamins act as coenzymes for different energy reactions. Okay. Here we go. So I'm going to show you a little video. Let's see. I didn't check the speaker. Let's see if the speaker works. Sorry.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
So you can see how NAD+ has to be used for this reaction to occur. Okay, so you see how the enzymes assist in the reaction, but we need coenzymes such as NAD+ and FAD+ for the reactions to occur. Okay? I mean, that's kind of the overview of how it works. All right. So when we talk about this reactions, we call it cellular respiration, where we oxidize food molecules to gain energy. All right. So we have two types of cellular respiration. One is aerobic, and when you see the word aerobic, you think of air, right? So it's going to require what? Oxygen to occur, okay? Versus an means without, right? Anaerobic means no oxygen. So let me kind of break it down how our body does this. Let's use an example. Say you're a walker. You like to go out and walk, right? So as long as you're bringing in enough oxygen during your exercise and the cellular metabolism of glucose, would you be doing aerobic respiration or anaerobic? Aerobic, all right? And in the aerobic respiration, you're using oxygen, and we're going to talk about how this works in oxidative phosphorylation reaction to occur. And you're making 30 to 32 ATPs. That's a lot of ATPs, isn't it? All right? Versus like you're a runner, you may not be bringing in enough oxygen for your cells to be doing aerobic metabolism, so you start converting to anaerobic metabolism, and you're making 2 ATPs. So which is more? Would you rather have $30 to $32 or $2? 30 to 32. So you can see in the metabolism of glucose, aerobic metabolism is very efficient. You're making a lot of ATPs, right? Versus anaerobic metabolism is kind of your backup system, not making that much energy in the form of ATP. But your body always has a backup system, doesn't it?
So are you saying it's better to walk than it is to run?
The question was, is it better to walk instead of run? If you're wanting to burn energy substrates, aerobic metabolism, yes it is, more efficient. Does that make sense?
Yeah.
You're like, oh, I see how this thing works. They said as long as you can sing and talk, right? Bringing in enough oxygen for aerobic metabolism to occur. Now, what we get with athletes, like runners and things, they do anaerobic metabolism will build up. Pyruvate gets converted into lactic acid, and we think it's that lactic acid that may make your muscles kind of sore. Now, they're kind of disputing whether it does or not, right? But we always used to say anaerobic metabolism, where you had a lot of pyruvate gets converted to lactic acid anaerobically, and anaerobic metabolism, it makes your muscles tired and fatigued and sore. Yeah. So you can see aerobic metabolism much more efficient, producing more ATPs, okay? And this is from glucose. All right, so here you go. What is aerobic metabolism? What is anaerobic metabolism? On page 289. What is aerobic metabolism? What is anaerobic metabolism? I could always remember these two, just their names. It helps me just to see their names and remember it. What is aerobic metabolism? What is anaerobic metabolism? All right, so now we're taking glucose, and through a catabolic reaction, we're basically breaking the bonds to capture those energies, those high electrons, and shuttling the energy with those high electrons with NAD that's going to be converted to NADH plus, or NADH plus hydrogen, and then FADH. So we're going to use this breaking the bonds to shuttle the energy into the mitochondria, where we're going to do oxidative phosphorylation. Okay, so glycolysis was the first step. So when you look at the words glycolysis, all right, let me get a pen real quick. We see the word, you take it, break it apart. I always break my words apart. All right, I didn't mean to mark through the L here, but we see glyco, which indicates glucose, right? So glucose. And lysis means to split. So we're splitting glucose apart, S-P-L-I-T. We're splitting glucose apart to make 2 pyruvate molecules, right? And in that process of glycolysis, we're going to make 2 ATPs. That's our net ATPs, okay? That's net. It took 2 to get the reaction started, and we end up actually making 4, but we only net two ATPs in this process of glycolysis. All right, and in that process, that energy that we have of breaking those bonds will help produce the electrons that are going to be shuttled into the mitochondria. If we only are doing aerobic or anaerobic metabolism, we're only doing glycolysis only. Okay, anaerobic is glycolysis only. What does anaerobic mean? Yeah, glycolysis only, without oxygen. If we have enough oxygen, then we're going to take these high energy substrates and we're going to go to this reaction, and then this reaction, and then this reaction through the electron transport chain, and these all occur in the mitochondria. The mitochondria of the cell. All right, and this is if we have adequate oxygen. So we're going to need oxygen for this reaction to occur. Because oxygen is going to be the last electron acceptor, and we're going to be, the oxygen bound with the hydrogen is going to be used to make H2O, which we know as what? Water. Yeah. So if we're only, this one occurs in the cytosol of the cell. And the rest of these reactions occur in the mitochondria. Okay? So if we don't have any oxygen, we're only going to do glycolysis in the cytoplasm of the cell with glucose. We're only going to make 2 ATPs. If we continue on through these reactions, we're going to make 2 ATPs here, 2 ATPs here, and then we're going to make about 28 ATPs here. And that's how we're going to get lots of ATP then through the electron transport chain. So you add it up, and we end up in the neighborhood of 32 ATPs, right? So you can see the electron transport chain is where we're going to make a majority of our ATPs, our energy that our cells are going to need, okay? But the weird thing about our cells, they only make the amount of ATP they need. So if we're not needing ATP for these reactions to occur, then we're not undergoing these catabolic reactions to make ATP. Our cell's only going to make what it needs. Y'all ready to go on? All right. So you can see glycolysis occurs in the cytoplasm, and then you can see the little bean-like structure in our cells called a mitochondria. Now, muscle cells have thousands of mitochondria. One muscle cell has loads and loads of mitochondria. Okay, so you can see where all the ATPs are made are in the mitochondria of the cell during electron transport chain. And you'll notice from going from glycolysis to that transition reaction, you'll see this guy, which is called acetyl-CoA. He's the branch point of this whole reaction. Okay, so if we're not needing ATP made, once we split into pyruvate, we go through this transaction, acetyl-CoA can be used to make fat, a fatty acid. All right. So if we're not needing energy, then the energy, the components, the compounds, acetyl-CoA can be used to make fat. Okay. Here you go. Here's your think time. So where does glycolysis take place in the cell? Where does the citric acid, also called the TCA cycle, happen in the cell? And where does the electron transport chain take place in the cell? This is all on page 289. So page 289 and 290. So where does glycolysis take place in the cell? Where does the citric acid cycle take place in the cell? And where does the electron transport chain take place in the cell? also, the citric acid cycle is also called the Krebs cycle, the TCA cycle. It has like 3 names. It's kind of confusing to students, I think. We learned it as the Krebs cycle, named after Dr. Krebs, right? Same thing. So if you get another class and they call it something else, you're like, oh yeah, that's the same thing. It's just called 3 different things. Citric acid, Krebs, TCA cycle. Okay, so where does glycolysis take place in the cell? Where does citric acid cycle take place in the cell? Where does electron transport chain take place in the cell? All right, so back when I was a student, we had to memorize all that stuff. We had to memorize the substrates. We had to memorize the enzymes. You guys don't have to do that. I feel like you can look that stuff up in a book. The main thing is this is showing you glycolysis, all the steps of glycolysis. You want to go back and learn it for yourself, get after it. I did. I had to memorize it. Now, if I had to sit down and write it out again, yeah, it'd be kind of iffy there on every single substrate and enzyme. But you can just see we're going from the top, glucose, making 2-pyruvate. There you go. That's the gist of the whole thing. And in that process, we're making 2 ATP, 2 net ATP, because it took 2 ATP to get started, and we end up with 4. 4 minus 2 is 2 net yield. All right. So this is how glycolysis works.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
By the way, the liver can take the lactic acid and convert it back into pyruvate in anaerobic conditions. So the good old liver has all the cool enzymes to do stuff, and that's called the Cori cycle. So the Cori cycle that can, anaerobic conditions, convert the lactic acid back to pyruvate, okay? So
Cells derive energy from the...
No, we're done. Keep going. It's not wanting to go. You're almost good. Come on. These little animations, when you embed them, sometimes it gets a little confusing. Come on, do it. I may have to stop it and go to the end. There we go, to get past it. All right, so this is just showing you the steps of the TCA cycle or citric acid cycle. I don't expect you guys to remember all the substrates, enzymes, and so forth anyway. But you can see in this process we're making 2 more ATPs. We're also breaking the bonds so that the hydrogens with their high energy electrons can be attached to the NADH, or NAD to form NADH and FADH2. So in this process, we're breaking these bonds, we're releasing the energy that's going to be shuttled in through the electron transport chain. Okay, and again, this is also taking place in the mitochondria of the cell. All right, so the process of transferring the electrons along a series of carriers is called oxidative phosphorylation, Again, this occurs in the mitochondria of the cell. And so we're using the energy in these substrates and these electrons, again, to phosphorylate ADP to make ATP. And we also have to have some minerals for these reactions to occur. Okay, so copper and iron are both required. So what if you don't have enough copper or iron in your diet? How are you going to feel? Everybody knows low iron, you're going to feel very tired, are you not? So you can see these guys are also very important in these energy metabolic reactions. So iron is called a cytochrome, and it's an important component of that electron system where the electrons are going to be shuttled across the membrane so that the energy can be captured for phosphorylation. Okay? Copper also a very important part of the enzyme reaction. So we have to have copper and iron for these reactions to occur. All right, so that's just showing you how the reaction occurs, kind of an overview. So we're using the energy captured from those electrons to phosphorylate ADP to make ATP. Okay? And that's just showing you the reactions. So are we yielding a lot of ATPs from oxidative phosphorylation? What do you think? How many ATPs are we making? From one molecule of glucose, we're making, do y'all remember? This one's the last one. We're making 28 molecules of ATP. That's a lot, isn't it? Yeah. So oxidative phosphorylation is where we make a majority of ATPs. This is just showing you one example of one ADP plus phosphate to yield ATP. And you'll see the oxygen is the last electron acceptor. We have to have oxygen for this reaction to occur. And we use the hydrogen binding to the oxygen to make water. All right, so here we go. Y'all ready? It's a little animation.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD plus and FAD are reduced to NADH plus H plus and FADH2. In the mitochondria, the electrons from NADH plus H plus are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
That was kind of a simplified overview to kind of get a feel for how it works. We're not going into heavy detail on this. You'll get more in physiology, but this is to introduce you to metabolism and how it works. Okay, so you can see we're making a lot more ATPs in the electron transport chain. Also called, what's the other name for electron transport chain? What do we need to have for this reaction to occur? What do we have to have for this reaction to occur? Oxygen. So it's also, and you're adding a phosphate group, right? ADP plus phosphate. So it's also called oxidative phosphorylation. Does that make sense? You have to have oxygen. We're phosphorylating, adding a phosphate group to ADP to make ATP, right? Thus you get oxidative phosphorylation. And we're transporting the electrons and across the membrane, so it's called electron transport chain. Does that make sense? Why it's named that? Once you get an idea of why something's named something, I think it makes more sense. And then it talked about those cytochromes. what do we need to make the cytochromes? You guys remember the mineral? We have to have iron. Yeah, and copper. Yeah. Copper is also important for enzyme reactions, but iron. All right, so we talked about anaerobic metabolism. We're doing this not in the mitochondria. For example, red blood cells don't have a mitochondria, right? So all their metabolism is what? Anaerobic, right? All their metabolism is anaerobic. So if there is no mitochondria, or if they don't have any oxygen, then they'll conduct anaerobic metabolism. And then we talked about the liver converts the lactic acid, also called lactate, back into pyruvate in something called the Cori cycle. All right, and this is just showing you going from pyruvate down into lactate. And then the liver has the enzymes then to convert the lactic acid or lactate back to pyruvate. All right, so now we're moving into fats. All right, so fat metabolism can only occur in the mitochondria. So what does that tell you? We have to have oxygen. All right, so in that process of breaking down these long hydrocarbon chains of fatty acids, we're breaking apart 2 carbons at a time, so it's called beta oxidation. All right, so the overall gist of this thing with AT production from fats is that we can make a lot of ATPs from fats. So one fatty acid, one long chain fatty acid. My writing is terrible with a little writer. I'm not getting better, I don't think. One fatty acid can yield 106 ATPs from one fatty acid. Does that remember many ATPs? 106. So we said with oxidative phosphorylation, how many ATPs can we make from glucose? Yeah. Well, overall, we're going to make about 32, right? One fatty acid can make 106 ATPs. Now, triglyceride is made up of how many fatty acids? 3 of these guys. So you can see why fat's such a great energy source, right? But in order to burn our fats, like exercise, we're going to have to bring in what? Oxygen for beta oxidation to occur. Because it only occurs in the mitochondria, it requires oxygen. So when we're out running, are we typically burning fats? No, we're using the energy substrates. We're going to talk about glycogen next week. That's used as the energy source. So we need oxygen to break apart the fatty acids for the energy for our muscles to contract. Okay, so this takes place in the mitochondria only. All right. So lipids or fatty acids cannot be converted into glucose. Just kind of heads up on that one. We'll get to that here in just a bit. We have a little shuttle that shuttles the fatty acids from the cytosol into the mitochondria, and it's called carnitine. You'll see a lot of energy drinks that have carnitine in them. Y'all have seen those before. Our body makes carnitine. We don't have to take it from an energy drink. Okay? So some people think it helps, but it's not required. Does that one have it? I bet it does. It's a Monster, one of those energy drinks. A lot of those exercise drinks, it does have carnitine in it. Is it required? No, your body can make it. All right. So things that help promote fatty acid oxidation, also called beta oxidation, where we're breaking those carbons apart, releasing energy 2 carbons at a time, is promoted by the hormone glucagon, growth hormone, and epinephrine. And again, we're getting a whole bunch of ATPs in this process. All right, and this is just showing you the fatty acid, the carnitine shuttle, the process of beta oxidation, and making acetyl-CoA. Once we get to acetyl-CoA, then we can go through oxidative phosphorylation and make a lot of ATPs. All right, so what is the process of making ATP from fats called, and where does it take place in the cell? And this is on page 296. 296. So what is the process of making ATP from fats called, and where does it take place in the cell? So what is the process of making ATP from fats called, and where does it take place in the cell? So the breakdown of fats to make energy in the form of ATP, and where does it take place in the cell? By the way, the term we use for splitting and breaking the bonds between these carbons, and breaking them 2 carbons apart of these long fatty acids is called lipolysis. So you get to physiology, see these terms, and you'll do them in your Learn Smart. So breaking apart the fatty acids in 2 carbons at a time, you see the term lipolysis. But if we broke this word apart, we'd do it right here, right? Lipo means, when you see lipo, lipids, you think of what? Fats, right? This is the easiest way to do terminology. And we see the lysis. What does it mean to lyse something? Split. So the splitting apart of these fatty acids, I just keep wanting to do spit instead of split. I'm getting ahead of myself. Splitting apart these 2 carbons at a time is called lipolysis. Sometimes you'll want to say lipolysis, don't you? Just looking at it, it looks like lipolysis. Yeah.
Okay, well, it's been a long time since I've been in chemistry, but this is like bothering me.
Why?
Acetyl-CoA. So acetyl is that alcohol?
Acetyl-CoA.
CoA.
Ah,
Acetyl.
you're thinking of alcohol metabolism. Uh-huh.
But acetyl, what is
Uh-huh.
acetyl?
Do you
Doesn't that mean
remember
alcohol?
acetyl-CoA from alcohol metabolism? We're almost there. Hold that thought. Hold it. That's a perfect thought to think of. Hang on, I'm getting ready to present it to you.
I just want to understand it.
I know, we're going to get it to you. Hang on. All right. So, well, let's just go ahead and do it. All right. So once we get to that acetyl-CoA branch point, right? Acetyl-CoA, also in alcohol metabolism, you can go 2 ways. You can either continue on to acetyl-CoA and make energy through oxidative phosphorylation, or the acetyl-CoA can then be converted into fat. It can be used to make a fatty acid. So keep that thought right there, okay? That's enough to hold you right there for a minute. We can run back just for a second. Okay. So, incomplete fatty acid oxidation is called ketogenesis, and we actually form ketone bodies. And you'll hear a lot about this. People on ketogenic diets, this is exactly what they're doing. So if we do not have enough carbohydrates in our diet, we will then start making something called ketone bodies from the acetyl-CoA. All right, so these ketone bodies, as they build up, makes the blood become acidic. All right, in normal ketogenic diet, you're getting quite a bit of ketones, but we're not to ketoacidosis, typically speaking, like you see with type 1 diabetes. If the ketones keep building up, because again, in type 1 diabetes, they're not producing what? Insulin. Insulin is the key that unlocks the door that tells the cells to take up glucose. So the glucose stays in the blood. So it's like the body is thinking there's no glucose coming in the diet. And so it starts breaking apart the fatty acids and making ketones from the acetyl-CoA. And for a very short period of time, your brain can use those ketones for a source of energy. Your brain likes to only use glucose only. But if there is no glucose, there's no insulin, even though the glucose is high in the blood, then the brain will use ketones for a source of brain energy, right? And the patient will have a real fruity breath because those ketones are like acetone, and then you can smell it on their breath. It smells like alcohol almost, okay? So that's a real telltale sign. But if it continues to build up in the blood, it'll lead to a condition we'll call ketoacidosis. And so when we chart it, it will be charted like this, DKA. Does anybody know about DKA? You ever see it on a chart? Patient comes in, chart DKA. All right, diabetic ketoacidosis. So this acidic blood will pull the potassium out of the cell. The electrolytes are going to be out of whack. They're also going to be very dehydrated, so they're going to have to put them on IV therapy pretty quick and get them on insulin. All right, but you see DKA, diabetic ketoacidosis. Okay, you know that this is a pretty serious condition.
So if they eat sugar,
If they eat sugar, it's just going to keep building up in the blood. The cells do not have insulin. Insulin unlocks the door of the cells to tell the cells to take up glucose.
and type 1 diabetes is only in about 10%.
Yeah, 5 to 10%. Okay. What can happen though is this ketoacidosis, it drops the pH of the blood, it downregulates the neurons in the brain, and they can go into a coma. Okay. Yes. You have a question, Tucker?
Okay, so I know people that are like,
Ketogenic diet.
yeah, that have been for like months.
Months and months. They don't go here. Because typically speaking, we don't see this in or people who are on those ketogenic diets.
And I know that they just like how you said that their electrolytes like drop. So like they drink like chicken broth because it's like high in sodium and I guess electrolytes.
Just sodium. That's all really are. You're getting rid of chicken broth.
And they like salt their food a lot.
Mm-hmm.
But I mean, like you said, it like.
This is with this down here. This is really serious where it's build up to cause the blood becomes so acidic that it'll pull potassium out of the cells.
Is there any way to cure that?
Insulin. Give them a shot of insulin. The cells will take up the glucose, right? And now the cells can use glucose as a source of energy, right?
What if they're an alcoholic and they're addicted to alcohol and they're addicted to drinking and
Well-
they develop ketoacidosis and
Well, it's going to be, I mean, like the pancreas is gone. You'll see that sometimes, pancreatitis with alcoholism. It's the same, you're going to treat it the same. Yeah, if it's DKA, you're going to treat it the same, right? Because you don't have enough insulin. With a ketogenic diet, they're not going to operate very well without any carbohydrates, right? You have to have some carbohydrates or your energy level is nil, right? So even though they're eating mostly protein and fat foods, they have to have some carbs or they can't go, right? So they're going to have some carbs just to be able to operate.
Like
They're not at this
5%.
level, right?
5% of their calories and carbs.
Yeah, yeah. So remember we talked about you need 180 grams of carbs,
and
right?
if you don't have enough carbs, then you can develop pancreas. Well, I had stones in my bowel ducts.
Oh, gallstones. Yeah, we talked about gallstones. Okay, so the thing too about ketones, we talked about when you use high protein diet. Remember that urea builds up. I mean, it draws a lot of water. It takes a lot of water to excrete that urea, right? All right, so you guys ready to go? All right. We'll also see you'll see buildup of ketones from semi-starvation and even fasting, but not typically to this level where the blood becomes so acidic. This one becomes very life-threatening. They can go into coma, okay? Can lead to death. All right, so that's just showing you what happens in ketoacidosis. Sorry, that was loud. All right. So again, our body has an adaptive response, but you're really going to watch out for this patient. It can lead to death. All right, so that's just the other steps on it on the ketones. All right, here you go. Think time. How are ketones made? This is on page 298. How are ketones made? ketones made. All right, so we also can use protein for fuel. How many kilocalories does protein have? 4 kilocalories per gram. So obviously it can be converted into energy too, okay? So in that process of protein metabolism, we've talked about this term before. Deamination, D means to remove, right? Anytime you see the word D, it means to remove. I could always just break my words apart and remember stuff. Amine, you see the word amine group? All right, so you're removing that nitrogen group, that NH2 group, and in that process that leaves you that carbon skeleton that you can break apart, right, to make ATP. All right, so you can break those bonds for energy, for making ATP. Okay, so we call it, first we have to get rid of the nitrogen group. Now remember, this NH2 forms with hydrogen to form NH3, which is also known as what? Ammonia. Is this a toxic substance? Very, right? Very toxic. And the ammonia then, your liver can convert into what? Urea. And then what gets rid of urea? You have organ gets rid of the urea? Kidneys, yeah. And in that process, it's going to take water. All right, so again, back on the high protein diet, same thing. Getting rid of that nitrogen group through deamination, remember, chopping off that nitrogen group. Then we have that carbon skeleton that can be used to make ATP, and breaking apart the bonds of carbon skeleton. All right, so we have some amino acids called glucogenic. Genic means genesis, right? So what do you think these amino acids can be used to make? They can be used to make glucose. Yeah. So they get converted into pyruvate. Once we get a pyruvate, remember glucose, glycolysis where glucose is broken apart into 2 molecules of pyruvate? We go in reverse now, right? So we put 2 pyruvates together to make glucose. So we actually can use some amino acids to make glucose. So in that process of like starvation, where you're breaking apart body proteins, some of those amino acids in our body proteins can be used to make glucose for our brain. Okay, and they're called glucogenic amino acids. And so you'll see your list on page 300 of your glucogenic amino acids. You guys may have them in your Learn Smart. I'm not going to test you over it, but asparagine, arginine, aspartic acid, histidine, glutamic acid, glutamine, isoleucine, methionine, proline, valine, and phenylalanine are all ones that can be used to make glucose. Now, not all amino acids can be used to make glucose. The ones that are called ketogenic, we see ketogenic, right? What do we think these guys are going to make? Acetyl-CoA. Acetyl-CoA can be used to make ketones, right? That's why it's called ketogenic. These guys cannot be used to make glucose, but acetyl-CoA can be used to make energy, right? And then the acetyl-CoA can also be used to make fat. Okay. So that's just showing you the process of glycerol from the fatty acids can be used to make glucose. and the glucogenic amino acids can be used to make glucose. Okay. So the process of making glucose from glucogenic amino acids is called gluconeogenesis. So gluco would be what? Glucose. Neo means new. Genesis means creation. In the beginning, right? First book of the Bible? Creation. So creating a glucose from something that's not a carbohydrate. Does that make sense? Yeah. So we can use the glucogenic amino acids to make glucose for the body. That's kind of handy because your brain needs glucose, your red blood cells need glucose. But fatty acids cannot be used to make glucose. But the glycerol part can be used. It's the only part of the triglyceride that can be used to make a glucose. All right. And that's just showing you the nitrogen group, the amino group, being made into ammonia and then urea and out the body from the kidneys. All right, here we go. So what materials can be used to make glucose in the process called gluconeogenesis? And this is on page 301. Gluconeogenesis. What can be used to make glucose in a process called gluconeogenesis? And by the way, this occurs in the liver, and some kidney cells can also do this. So that's kind of cool. So what substances can be used to make glucose in the process of gluconeogenesis? All right, now we're to alcohol again. All right, since you brought it up, we mentioned it. So, lo and behold, what did we find out a while ago? Can alcohol be converted into acetyl-CoA? It surely can. Yes, it can. All right. So we have that enzyme, alcohol dehydrogenase, and it can convert alcohol into acetaldehyde, which is toxic, and that acetaldehyde then can be converted into acetyl-CoA. Right. And if we're making gobs and gobs and gobs of acetyl-CoA, if you're drinking a lot, that acetyl-CoA, you only need so much energy. The acetyl-CoA then will be converted into fat. That's how fat chokes the liver cells and leads to cirrhosis of the liver. Okay? And we talked about that before. So that's just showing you how it happens. There's your answer, Ellen, right? Mm-hmm. And thus we get to acetyl-CoA, and we end up with fat. Acetyl-CoA can go right into be making a fatty acid. Okay. So the liver has the coal enzyme, so if you destroy your liver, you're in trouble, right? You don't want to do that because it has all these coal enzymes to do some very important reactions. In our body's ATP on our needs, we only make what we need. All right, so high ATP makes anabolic reactions. High ADP is going to make ATP, or catabolic reactions that we need. And it's stimulated by enzymes, hormones, vitamins, and minerals. Okay. So this is kind of the overview. If you'll notice from this, though, look, everything going down to make acetyl-CoA. Woo, here we are, acetyl-CoA. The big branch point of this whole thing. You guys paying attention? This is like your overview for physiology. So look, carbohydrates, alcohol, lipids, proteins can all be used to make acetyl-CoA through catabolic reactions. And once we get to this acetyl-CoA, we can go on, and if we need energy, we can go through the electron transport chain and make energy for ATP, right? Or if we don't need any ATP, we go in reverse, all right? So the acetyl-CoA can be used to make fat.
And so that CO2, carbon, oxygen, and what does that 2 mean?
CO2 is two oxygen to a
So
carbon.
carbon,
Mhm.
2 oxygen plus hydrogen, plus 2 hydrogen, plus, or oxygen plus energy?
Mhm.
Is that how that is read?
Carbon dioxide, right? Carbon dioxide plus water plus energy in the form of ATP.
CO2
These are
is-
all the carbon
Or H2O
dioxide.
water,
Huh?
yes.
Carbon dioxide. This is all the end products of metabolism. So you can see, look at all the vitamins and minerals you need for these reactions to occur, though. Look at all these B vitamins. What? We need a whole bunch of them, don't we, for these reactions to occur.
So if you take a multivitamin, would that cover?
Would that help if you're taking a multi? Check the label, see what's in them, right? But you can see you're needing a bunch of B vitamins because they're acting as coenzymes for these energy reactions to occur. We also talked about minerals. Can you remember any minerals we need? Copper and iron for these reactions to occur. Okay, but you can see lots and lots of B vitamins. You'll also see, like, in this reaction we need magnesium, and in this reaction going in reverse to make amino acids linked together to make protein, we also need vitamin C and vitamin K. So you can kind of see how we need the B vitamins in particular for energy metabolism. But once we get to this point, you can see carbohydrates can be made to acetyl-CoA. If we don't need the energy, they go in reverse and we make what? Triglyceride, right? Which is fat. If we get too much alcohol, acetyl-CoA, we're making triglycerides, we're making fat. We get too much protein, we're going down here making acetyl-CoA, too much acetyl-CoA. Now this is going to take some energy to break it apart. So this is the least method that's used. These two, it's pretty easy, especially this method. Breaking apart lipids to acetyl-CoA, it doesn't require a lot of energy. We can make fat, fat, fat. So if you eat a fatty diet, you can see too many calories of any type, it's going to lead to stored lipids. Does that make sense? Whether it's carbs, lipids, or proteins. Okay. All right, so the good old liver, look how much stuff the liver does. Makes fats and cholesterol, converts the monosaccharides, such as fructose into glucose, galactose into glucose, amino acid metabolism, ammonia to urea, alcohol metabolism, nutrient storage. That's just a few of the jobs, okay? Has those great enzymes for the reactions occur. And this is where everything occurs at, too. And this is like the overview. Take this one image, it's on page 305 in your book. When you go to physiology, you can go back to those pages. This is super helpful because it's the whole big picture of everything going on. Okay? So it's super helpful. Put a little mark in your book for physiology. All right, so fasting, when you're not eating, encourages breaking down glycogen. By the way, what is glycogen broken down into? Glucose. And what is this reaction when we're breaking down glycogen? What do we learn the term today? It's called, which one? Breaking apart glucose is glycolysis. But the whole reaction is called? Almost. Is it anabolism or catabolism? Catabolism, yeah. Catabolism. Absolutely. Isn't that cool how you already know stuff, right? You're breaking apart, right? So glycogen's binding glucose together. Breaking apart triglycerides and proteins into amino acids would also be catabolism. Gluconeogenesis, which is what? What is gluconeogenesis? Making glucose. Ketogenesis, making ketones. Urea, a byproduct from, yeah, amino acid metabolism. See how you know all these things now? This thing, of course, is going out in the urine, right? Yeah. So this is what feasting encourages, making glycogen. So we're taking glucose, linking it together, right, to make stored glucose in the form of glycogen. So what's this reaction called, going from glucose to glycogen? Anabolism, yeah. Did the catabolism thing I helped you guys with earlier, talking about cannibalism, breaking apart big stuff into a little piece at a time. Does that help? It always helped me.
I just think of like anabolic steroids.
Anabolic, yeah, building up muscle. There you go. All right. So same thing. We can take our 3 fatty acids plus glycerol, right? We can take acetyl-CoA, make 3 fatty acids, add glycerol with it, and make triglycerides, right? Oh, sorry, my writing's bad. Okay, all right, lipogenesis. You see, lipo is creation, or genesis is creation, lipid creation of fat. See how that works? So we're going the other way. So if we're eating, we're going this direction, right? We're storing up. If we're not eating, we're going the other direction. Simple as that. Same thing. Taking amino acids, making proteins, going the other way. All right, here we go. What nutrients can be made into acetyl-CoA? And if you didn't pay attention, there's a little thing on page 307 that can help you, a little figure that we had a while ago. What nutrients can be made into acetyl-CoA? What nutrients can be made into acetyl-CoA? Isn't it cool when you already know this stuff now? You can do it without looking, I hope. At least kind of get a feel for it. So what nutrients can be made into acetyl-CoA? All right, and that's just where we're storing in the fat tissues, right? How much energy it requires and how efficient it is for the body. Inborn errors of metabolism, page 311. We talked about phenylketonuria that builds up the phenylalanine. This is a genetic disease. All these are genetic disorders. And so remember, phenylalanine is an essential amino acid, so they just have to go on a lower phenylalanine diet, including a special formula. And we said they have to limit the phenylalanine in the diet for the rest of their life. And then tyrosine becomes a conditionally essential amino acid at this point. So even, we talked about Diet Coke, contains phenylalanine from aspartame, the artificial sweetener. In galactosemia, you see galactosemia. What is galacto? You see something you recognize with galactose? Unable to metabolize galactose, right? Where do you think we'd find galactose?
Lactose.
Yeah. So you're thinking of dairy. Are they going to have to eliminate dairy? Absolutely. Another genetic disorder, very serious. The first one, of course, can cause mental retardation, failure of the child to grow. Second one, same thing, mental retardation, bacterial infections. So they're going to have to limit any of the dairy in their diet. Glycogen storage disease, this one also is very interesting. They have the inability to convert glycogen into glucose. And so they have to eat raw cornstarch continuously at pretty regular intervals to help to maintain the amount of glucose circulating for the brain. Okay, so the first one's the most common of these inborn errors of metabolism genetic disorders. All right, you ready for a think time calculation? I'm going to help you on the first one, and then you guys are on your own on the next ones, and then I'll stop the tape. So I'm going to show the online students how to do this one. Okay, she's a 160-pound female. She's 5'3". Okay, there's two methods we can do it. The simplest method, I told you guys, is not doing the conversions. You can do pounds to kilograms and inches to meters, or you can use the other formula, which uses pounds and inches. So we're going to do that one, okay? So she weighs 160 pounds. So it's 160 pounds. My writing with the markers very hard on mine. All right. And it's times 703.
Wait, where did you get 703?
This is a formula.
Okay.
This is a formula for calculating BMIs. And divided by inches times inches. All right, and what were her inches? She's 5'3", so she'd be 63 inches. Good. 5 times 12 is 60, so it'd be 63 and times 63. All right, so you do the top part first, 160 pounds times 703, and then you do inches times inches, okay? So someone do the math for me real quick. 160 times 703 is
112,480.
100, how much?
112,480.
That way you guys at home can know how to do this too. Divided by 63 times 63. 3969. All right, and so her BMI is is 28. All right. Is that what you guys got? Everyone got 28? All right. So now we can look at our table in the next chapter. And for each of these patients, I want you to tell me if they're in a normal body mass index and if they're an overweight or obese category. Now, BMI is not end-all, do-all, you guys. I just want to let you know that right up front. But it is, a lot of doctors use it, and it is a way to help to determine maybe if they're in overweight category. So if we look our table on page 329, is a 28 BMI in the normal range, a healthy range, overweight, or obese category? What do y'all think? Yeah, she's in overweight category, yeah.
According to BMI, it's on mine.
Yeah.
I'm like 28 too.
All right, good deal. All right, I'm glad you brought that up. We're going to talk about that next week. So I have a bodybuilder. He's doing a lot of bodybuilding. We see this a lot with athletes. Arnold Schwarzenegger, when he won Mr. Universe, had a BMI of 40. It was all lean muscle mass, right? So it's not the end-all, do-all. All right? Waist circumference is a better example. All right? But I'm just saying doctors use it a lot. You guys need to know how to do it. BMI is not the end-all, do-all, but you need to know how to do it. And you'll see those charts on the doctor's doors. So just so you know how to do them. Nurses use them all the time to you, and you're going to be calculating. So you guys do the rest of the patients, right? Yes, please round, and these are all your think times. So you guys do the rest of them, okay? Y'all have any questions on how to do these for your homework? It's not hard, is it? Not hard math. So I'm going to stop the video.